The University of Leuven is the largest university of Belgium. It has about 40,000 students and is a very strongly research-oriented university, also very strong in innovation and tech transfer. VIB is the Flemish Inter-University Institute for Biotechnology. It has about 8 centers that are located at the different Flemish universities and also focused on fundamental research and on tech transfer. And we are working in the Department of Biology of the University of Leuven and in the Center for Microbiology of the VIB. The main goal of our research is to elucidate the mechanisms by which nutrients are sensed by cells. We want to identify new mechanisms by which nutrients can regulate the growth, the metabolism, and other physiological properties of cells. This overactive sugar breakdown in the cancer cells is called the Warburg effect. And up to now it has remained unclear whether this Warburg effect is just a symptom of the cancer cell or whether it could also be causative, whether it could stimulate the aggressiveness of the cancer. And we use yeast cells as a model system for that because yeast cells are experimentally very easy to use and it's also a genetically tractable system which gives very strong advantages compared to the use of cells of higher organisms. Yes, both yeast and cancer cells have the unusual property that they prefer fermentation over respiration. They prefer to ferment sugar into ethanol or lactic acid rather than respiring the sugar to carbon dioxide. We have now identified a mechanism by which the very active sugar breakdown in cancer cells can stimulate the aggressiveness of the cancer. And this seems to support the data in the scientific literature that sugar-poor diets may help the patient to overcome the cancer, may help the positive outcome of chemotherapy.
One of the key moments in our research for sure was the collaboration with the people of the Structural Biology Lab in Brussels, the VIB. We were looking to confirm our hypothesis that we formed about the interaction of sugar, phosphates and RAS in yeast cells, to see if this hypothesis would also be true in a mammalian context. Activation effect was observed, and remarkably in the same physiological range as we had observed in yeast cells.
We really asked the question, how can we move forward from this? So we have the strong phenotype, we have the influx of glucose, we have the overactivation of RAS. We decided to use this strain, specific strain, to screen compounds against. Because if we can find a compound that would counteract this phenotype, it can have potentially immediate important effects if we would go to mammalian cells and specifically go into cancer cells.
Our research really focuses on cell signaling diseases that are characterized by aberrations in protein phosphorylations and dephosphorylations. And cancer is, per definition, a typical example of such a signaling disease. What our role in this work was essentially to figure out whether RAS activation by glucose that was originally discovered and completely mechanistically worked out in yeast, whether that mechanism was actually conserved in human cells. The initial challenge was to find ways to culture the cells in the absence of glucose. The solution was also partially based on the work that was done in yeast, and the solution turned out to be to grow the cells in the presence of galactose. In my opinion, the medical relevance of this work is twofold. The precise molecular elucidation of the mechanism by which a glucose metabolite stimulates cancer cell growth clearly holds a therapeutic potential. The second implication of this work is maybe a little bit more controversial and has to do with the role of nutrition, the role of a specific diet that a cancer patient gets during his or her treatment. We get these high glucose peaks in the blood, for instance, immediately after a normal meal, because it is these high glucose levels that might do a lot of harm and that might actually maintain tumor growth. Unfortunately, this is not yet sustained by a lot of clinical studies. And I do hope that our work now would give a kind of incentive to the clinical oncologist community to keep this idea in mind and to set up dedicated studies to answer that question.
Well, the research in the VIB will now be oriented further on this Warburg effect. We have now explained why the Warburg effect is important for cancer, but we do not know yet the primary cause of the Warburg effect. And we think that this primary cause is also conserved in the yeast cells. So we are concentrating now on the elucidation of the very first mechanism that triggers this Warburg effect. When we have identified this mechanism and we think that it's specific in the cancer cells, then we can screen for drugs that would specifically counteract this effect and make the cancer cells more sensitive, for instance, to chemotherapeutic agents, which would facilitate the combating of the cancer.